weekend? 100%.
Nope. I returned.
Nope. I told him. And it was like, yeah, I know. So stapled mine together. Probably.
Okay, so by way of introduction, we have looked at the biochemistry, the composition of protoplasm. We put a membrane around that protoplasm. Then we talked about eukaryotic cell structure and function, and then prokaryotic cell structure and function. And then we talked about taxonomy, which is this business of categorizing things, trying to put organisms of similar types into groups together. Now we're going to start looking at the groups. So we're going to start looking at prokaryotic organisms. Tonight we're going to be looking at archaea and bacteria. And then we'll go through the fungi, the protozoa, the microscopic algae, and the multicellular parasites that usually relate to microbiology. So tonight is prokaryotic cells, again, archaea and bacteria.
That's according to the light.
So in the mid to late 1980s, the prokaryotic organisms were categorized into 4 volumes of what's recognized as the primary reference for identification of bacteria. It's called the Burgess Manual. There was a Burgess Manual Determinative Bacteriology, and then there's a Burgess Manual Systematic Bacteriology. I'm going to talk more about these in the laboratory. But the 4 primary groups were ordinary gram-negative, ordinary gram-positive. In those 2 groups alone, almost all the clinically significant organisms would have fallen. Then there were bacteria with unusual properties. Some of those included spirochetes because they're unusual. They have the paraplasmic flagellum. And some of the other organisms that were unusual went into that category also. Bacteroidetes and Chlamydia. And then the last group were the filamentous bacteria. Well, that's not that long ago, mid to late 1980s, but this thing has been completely revised. So this was the first Burgess Manual of Systematic Bacteriology. Then they did a second volume, a second set of Burgess Manual of Systematic Bacteriology, and now they've done the whole thing again. Fortunately, now they have finally put it online, so they can begin to try to keep up with the new information that's being generated relative to prokaryotic cells. So there's a tremendous amount of information. Most of the evidence we now have for the huge volume of different types of bacteria comes from looking at nucleotide sequences, specifically ribosomal RNA nucleotide sequences. The most important one for bacteria is the 16S. They also have 5S and 23S. When Carl Woese compared prokaryotic cells, he found out they were in 2 completely different categories. And so the R.H. Whitaker kingdom Monera disappeared and was eliminated. You can't have 2 extremely diverse groups of organisms in the same kingdom. So the kingdom Monera disappeared, and we have 2 separate domains, the archaea and the bacteria. The domain is a level of classification above the kingdom. As far as I know, there still is no kingdom for prokaryotic organisms. But anyway, so we're going to look at these 2 groups. You'll see lots of representations of the 3 domains of life. So the 3 domains are archaea, bacteria, and eukarya. Everything eukaryotic is in the eukaryotic domain. When you see illustrations like this, keep in mind that although this looks nice on paper, it is highly inaccurate, because this group is much, much smaller than this group. And this group is huge, absolutely huge. We know relatively little about it, although we're going to go through it tonight, and some of you are going to go, really? Do we have to know all of that? And yet it's maybe somewhere between 1 and 10% of what's out there. We really don't know. We know very little about bacteria. So we're going to start with the domain archaea. When I say we know relatively little about bacteria, perhaps we know even less about these guys. The archaea. So this is a domain, if you take a general biology class, it may not even be mentioned. These things used to be called archaeobacteria. Arch means ancient. They were classified originally as bacteria with unusual properties. This is before Carl Woese did his investigations using the ribosomal RNA, and Woese demonstrated again that they were not just unusual bacteria, they were not bacteria at all. So their nucleotide sequences are very, very different. We're going to find out there are other reasons why these things are not bacteria. They look kind of like bacteria. So when you see these electron photomicrographs, you see cells that look like bacteria. You see rod-shaped cells and galaxy-shaped cells. Some are in clusters. So they do morphologically resemble bacteria, but they're not. So why are they not like bacteria? Well, first of all, their cell walls never contain peptidoglycan. So peptidoglycan is the rigid material that makes up the cell walls of most bacteria, or I should qualify that and say most bacteria that we are familiar with. It's the rigid material. We talked about peptidoglycan when we were doing the ground stain. So archaea never have peptidoglycan in their walls. They do have a material that's called pseudomurin. Murin is another name for peptidoglycan. So they have a molecule that's somewhat similar, but it's not the same stuff. This is showing the archaeal cell wall. So here's the plasma membrane, cytoplasmic membrane. This pseudomurin layer is out here, and this is about as vague as you can get for an illustration. And then outside of that, there's an S layer that's primarily proteins or glycoproteins. Perhaps somewhat similar to the gram-negative outer membrane, where you have that layer, a very large number of lipopolysaccharides out on the surface. Well, these are glycoproteins, not lipopolysaccharides. Anyway, the other thing about the archaea is that their cell membranes are very different. So when we talked about the composition of cell membranes, where you have a lipid bilayer that's made out of these phospholipids primarily, the phospholipids have hydrophilic heads, hydrophobic tails. And those fatty acids that make up the phospholipids are attached to a glycerol molecule. It turns out that the archaea have a very distinctly different glycerol in that it is a mirror image molecule. So if you look at bacteria, they have L-glycerol. No, they have D-glycerol, excuse me. So the archaea have L-glycerol. It's a mirror image molecule. It's kind of hard to recognize this when you look at the diagrammatic representations. But here's glycerol of archaea. This is the glycerol of archaea. This is the glycerol of bacteria. So it's a mirror image molecule. The other difference is that in bacteria and eukaryotic cells, the lipids attach to the glycerol, so you have two fatty acids attached to glycerol, and they're connected with ester linkages. In archaea, the link between the lipid and the lipids and the glycerol are ether linkages. So the linkages are different. The other thing about them is that the archaeal cell membranes have these fatty acids that aren't really, they're branched isoprene chains, and they have side chains coming off of them, and sometimes they have ring form molecules associated with them. So they're really different in terms of cell membrane composition. The other thing that's shown in this illustration, some of the lipids associated with the archaeal cell membrane go all the way across. So they've got a phosphate group here and a phosphate group here, and these lipids are attached all the way across. So instead of having a bilayer, you have a monolayer. They don't all have that, but some of them do. So generally, cell membranes of archaea, very distinctly different. Different glycerols, different types of lipids attached, different type of bond holding them on. And then the third thing is the nucleotide sequences. Again, if you look at the ribosomal RNA genes for archaea, primarily the 16S ribosomal RNA, if you compare archaea and bacteria, you find out that those nucleotide sequences are not the same. The other thing about archaea, at least some of them, they do have histone proteins. Typically, bacteria don't have histones. So we talked about the composition of chromatin in eukaryotic cells. The DNA is wrapped around the histone proteins, and then the histones come together to form the chromatin. The archaea are more similar to eukaryotic cells in that respect. Also because archaea at least sometimes have introns. Apparently bacteria don't. So introns are the segments of DNA that end up not coding for protein formation. Introns get cut out and exons get connected together. That involves spliceosomes. I mentioned spliceosomes when talking about eukaryotic cell structures, but we'll talk about them more when we get into the genetics section. So there we see histone proteins and introns that get cut out, and then exons get hooked together. And then, as I said, their nucleotide sequences in their ribosomal RNA are not like bacteria. Bacteria and archaea both have 70S ribosomes, and the 70S ribosomes have subunits that are 50S and 30S. The 50S unit has two pieces of RNA associated, and then the 30S unit has one. But if you look at the RNA types that are in prokaryotic cells, they're identified based on Sedberg units. They're 5S, 16S, and 23S. Both bacteria and archaea have those three types. It's the nucleotide sequences that are different. As I said, like words are different from one another because the letters are different. So one of the things that you'll find somewhat confusing when you're looking at information about archaea, because they were originally classified as bacteria with unusual properties, some of the names for the major groups of archaea begin with bacteria. So they have a prefix that is bacteria. The other thing is some of the substances that are produced by these organisms, the name begins with bacteria because people thought they were. Those kinds of names don't change. They're going to stay there, even though we now know that these organisms are not bacteria at all, they're archaea. So Methanobacterium, that's a type of archaea. Halobacterium, that's a type of archaea. And the substance Bacteriorhodopsin isn't made by bacteria at all, it's made by archaea. That's not confusing. Could we change those names? No, probably not. Again, this is an area we don't know that much about the archaea, and for a class like this, it's just an introduction. We're going to look at three major groups of archaea. There are other archaea, there's lots being found out about these guys. So our first group are the methanogenic archaea, and there are two classes, Methanobacteria and Methanococci. For you guys, don't worry about classes. I'd like you to, throughout this section, pay particular attention to terms that mean something. So methanogenic, what does that mean? It means they're capable of producing methane. Apparently the archaea are the only organisms that are capable of producing methane. Although if you read on the internet, you'll find out cattle produce lots of methane. It's like, no, they don't. The cattle happen to have symbionts living within their guts. And the symbionts, which are archaea, produce methane. It's not the cattle at all. At any rate, the methanogenic archaea produce methane as a waste product. And they do this by using carbon dioxide as an electron acceptor. And so they're going to be binding electrons and hydrogen protons, essentially hydrogen, to carbon and making methane from carbon dioxide. So methane, well, we know it's an important air pollutant, primarily because it's a greenhouse gas, so it's a considerable concern. But it's also a potential energy source because it's flammable. You can burn methane, and so you can use it for a variety of things. People are recognizing that where you have methane being produced, it is a potential energy source that we might be able to burn the methane, use it to heat water, maybe to drive turbines, to generate electricity, that kind of thing. There are methanogenic bacteria living in our gastrointestinal systems also, and they get deposited in wastewater treatment facilities. And so what's happening, kind of in modern wastewater treatment facilities, they used to just burn it. The methane that was generated in association with human waste was just burned. They'd have these pipes going off. If you go to the wastewater treatment facility in Roseville, the last time I was there, they were still just burning it, some of it. But some of it they were actually putting to use, because if they burn it and use it to heat water, then they can use it to heat facilities, and so they can maintain specific temperatures in some of their treatment facilities. And of course, you could use it to generate electricity, because you can generate steam and then use steam to run turbines and so forth. It is somewhat problematic. There have been instances where organic material that was buried, as the methanogenic archaea begin to break that down, they produce methane, which then comes to the surface, comes out of the soil. There was a period of time where there was a bike trail between Rockland and Roseville. If you rode your bike back there, there were signs that said, "Don't smoke in this area. Don't ignite any kind of material, because there's enough methane coming out of the ground it might catch on fire." There have been instances where people's basements have filled up with methane because their homes were built on landfills and the methane's coming out of the ground. Or you'd be in the backyard, light your barbecue, and your lawn catches on fire. Not a good situation. But anyway, so we're trying to harness that, recognizing that the methanogens are breaking down organic material. Actually, they live in association with other types of organisms that are breaking down the organic material, and then they're using the CO2 produced from that to generate methane. If you go out to the landfill now that's out, if you go out toward the casino, Thunder Valley, there's these big signs out there that say casino this way, landfill this way, and what you'll notice are these very large areas with pipes running on the surface. Well, they're generating methane from the buried organic material, and they're capturing that methane because it is a potential energy source. But when it's associated with cattle, this is a bit of a concern, because we have very large human populations now that like to eat meat. In this country, a lot of the meat is cow. In other countries, a lot of the meat is sheep or goat. These organisms are all ruminants, and they have archaea living in their gut. And again, the archaea produce quantities of methane. So I just looked this up this afternoon. About 25% of what's considered anthropogenic, that is methane generated in association with human activities, about 25% of it comes from ruminants, from us raising cattle, sheep, goats, etc. These animals have the archaea in their gut that produce methane, and they belch and release the methane, which has a significant impact on the atmosphere, because, as I said, methane is a greenhouse gas. So it's warming our atmosphere. So what do we do about it? Eat plants. At some point, we're going to have to stop raising cattle in large quantities, because we're going to cook ourselves. Okay, next category, the extreme halophiles. The class is Halobacteria. Again, don't worry about the classes, but you need to know what a halophile is. This was in our set on taxonomy. What is a halophile? They love something. Phil means love. What do they love? Salt. High salt. Halophiles are organisms that like high salt. And if they're extreme halophiles, they really like high salt. So these are gram-negative archaea. They live in salt, very high salt, 17 to 23% salt. They can live in water environments where the salt concentration is so high, the salt will be solidified on the top of the water, and there'll be crystalline salt down underneath. And they can still survive in there. So if you go to Utah, there's places near the Great Salt Lake where you can find these organisms. If you go to San Francisco Bay, where they're evaporating seawater to produce salt, you can see them. These organisms, there's a group in the genus Halobacterium. Again, they're archaea, even though the genus name is Halobacterium. They produce a substance called bacteriorhodopsin. Here you can see the bacteriorhodopsin. It's pink. So these are aerial, well, three of them are. Two of them are aerial images. One is just an image from the side. I think the two in the middle are from San Francisco Bay. So this is the pink color that you see here is due to the bacteria rhodopsin that the organisms produce. Here you see them growing on the plate, and this, I believe, was taken in Baja California, where they have salt evaporation ponds that are kind of in a more natural setting. At any rate, bacteria rhodopsin. This is the pigment that these organisms produce. What's rhodopsin? Anybody? Have you ever heard of rhodopsin? Seriously?
Yes.
Yes. Where is it found?
No, I said no, and I
No?
was agreeing to yes, I never heard of it.
Never heard of it. Wow.
Yeah.
Okay, well, rhodopsin is a pigment that's found in the human retina. It's one of the pigments that allows us to see. So this is a related protein, bacteria rhodopsin. Again, it might better be called Archaea rhodopsin, but the name is not likely to change because it goes way back in the literature. At any rate, it's similar to rhodopsin that's found in the human retina. Consequently, scientists are very interested in bacteria rhodopsin. It's something that they can work with, they can study to try to understand how rhodopsin works. It has been used to make artificial retinas, and it's even been used to design some special computers that take advantage of the fact that this protein, bacteria rhodopsin, changes configuration when it's exposed to light. So it is a membrane protein. When light shines on it, it changes its configuration. When it's in the dark, it goes back again. So it's kind of like having a 0 and a 1, which is part of the binary code for computer technology, and I don't know anything more about it. But at any rate, bacteria rhodopsin apparently can respond to light and can be used in computer technology. What the bacteria do with it, they use that change in configuration to move hydrogen protons across a membrane. So bacteria rhodopsin responds to light. When the light strikes it, it pumps hydrogen across the membrane, and this generates a proton gradient. If you have a lot of protons on the one side of the membrane, this is referred to as a proton motive force, because those protons don't want to stay there. They want to go back across the membrane, and they can go back across through an enzyme called ATP synthase. So in this diagrammatic representation, this is ATP synthase. The hydrogen protons go back through that, and that hydrogen flow is connected to ATP synthesis. So the bacteria rhodopsin is used to move the hydrogen proton across the membrane, generate the proton motive force, and then when the protons come back across the membrane, ATP is made. So the archaea are using this to generate ATP. And it is photophosphorylation because the energy initially came from light. The third group of archaea are the thermoacidophiles. There's quite a number of classes in this group. Here again, what's a thermoacidophile?
It loves acidic, hot.
Yeah, so they like acidic and warm, at least warm, maybe hot, warm to hot. So technically, thermophile is warm, hyperthermophile is hot. But these guys like warm, acidic environments. They're going to grow best there. So they live in some rather unusual environments where you have hot acid, probably hot springs. This picture is a thermal vent on the seafloor. And so you see that it looks like underwater smoke coming out of here. There's quite a lot of heat. So these organisms are of interest because humans can take advantage of enzymes produced by these organisms. We have a lot of industrial processes where in order to produce certain kinds of things, the enzymes have to be able to withstand heat and sometimes acidity. So people are interested in these thermoacidophiles to find out what kind of enzymes do they have and what could we maybe do with them. So there are now enzymes in detergents, enzymes used in textile production, food processing, paper processing, etc., etc. Again, this is quick. That's all I'm going to tell you about archaea, which is not very much. So now we're moving to the next domain, the domain bacteria. Within the domain bacteria, there are now multiple phyla, and for a period of time in the Burgess manual, these phyla were listed by number. The numbers are Roman numerals. So what you're going to notice, if I have a number in front of a phylum name, you'll notice I don't have all the phyla. So we start not at phylum 1, but out there a ways. never mind those numbers. That's not something you need to know. You do need to know the name of the phylum. So you need to know the phylum Deinococcus thermus. Don't worry about the fact that it's phylum 4. It's like phylum 4 according to who? Never mind. So, Deinococcus thermus. This is a phylum that contains two major groups, two genera of considerable importance. So again, we're talking bacteria now. We've left the archaea behind. The Deinococcus thermus are bacteria. And Deinococcus is of interest because these organisms are highly resistant. Resistant to heat, resistant to chemicals, resistant to radiation, resistant to desiccation. Apparently they're resistant to higher levels of radiation than anything could have ever been exposed to on the surface of this planet. So investigators are somewhat wondering how did they get like that? Why are they so resistant? They apparently contain enzymes that can repair damage to their DNA. One in particular, Deinococcus radiodurans, is a polyextremophile. So these are organisms that can survive extreme exposures, again, heat, desiccation, radiation, one of the toughest types of bacteria known. And so there's a common name that's sometimes applied, Conan the bacterium. That's Deinococcus radiodurans. So the Deinococcus genus, it's kind of interesting because we have isolated Deinococcus in this laboratory a number of semesters. And it used to be, if you look in the Berkeley's manual, there were 2 or 3 species of Deinococcus. Now there's 50 some odd. Lots of interest in Deinococcus. People want to know more about these things because they are radiation resistant. So why do we care? Well, if that radiation resistance has to do with enzymes that can repair damage to DNA, if we could isolate those enzymes, it might be possible that we could come up with something to treat humans that have been exposed to radiation. Right now, if you're exposed to radiation, you're pretty much a goner. There's not much that can be done for you. But it might be possible to use Deinococcus enzymes as therapeutic agents to repair damaged DNA. Okay, the other half of that phylum, Deinococcus thermus, is the genus Thermus. And we know thermus, if you think about thermophiles, these are organisms that like warm environments. The thermus are actually hyperthermophiles. They like hot environments. So these things tend to live near the hydrothermal vents on the seafloor, but also in hot springs. And there's one of particular interest to us. You can see they look kind of like bacteria. This shows environments where you might find the vents on the seafloor for hot springs. The one that we work with in the laboratory actually did come from hot springs in Yellowstone. The organism is called Thermus aquaticus. And Thermus aquaticus is the source for the enzymes that are used in the polymerase chain reaction, or PCR. So we're going to be using enzymes from Thermus aquaticus. The enzyme is called Taq polymerase. Polymerase is an enzyme that builds DNA, and this Taq, T is thermus, AQ is aquaticus. So it just tells you where that enzyme came from. Apparently, Terry Mullis, who was the individual that developed the modern day PCR using the enzymes from Thermus aquaticus, was accused of stealing the bacteria from the national park. I challenge anybody to visit a national park and not steal bacteria. Because you walk away from, especially Yellowstone, if you go out there anywhere near the geysers, you get into that mud and it's on the bottom of your shoe, and then it's in your car and you're taking it home. Because the bacteria go with you and you don't even want them. But anyway, he actually did want them, so he got them out of the hot spring there and then developed the PCR using this enzyme, which is heat stable. The significance of that is the PCR involves heating the DNA to denature it over and over and over again. You have to have an enzyme that's heat stable, and the enzyme from Thermus aquaticus was quite heat stable because they normally live in near boiling water. Okay, the next phylum we're looking at, notice we went from phylum 4 to phylum 10. What a phylum in between? Never mind. And you don't need to know the numbers. You do need to know the phylum Cyanobacteria. This is an interesting group. For a while, they were considered to be a really major, important group of bacteria. They are unique. The cells are generally larger than typical bacteria. they used to be called blue-green algae. In fact, we have these little booklets up here in the cupboard about, get the right one, how to know the freshwater algae. And if you look in these books, you'll find cyanobacteria. They are not algae. Cyanobacteria are not plants. My husband was showing me a photograph that was taken of a U.S. Forest Service sign that was a picture of a pond that had a lot of cyanobacteria living in it, and they had all this information about the unique plants that live in that pond. It's like, those are not plants. Those are bacteria. That's a different domain. The bacteria are not plants. So cyanobacteria, they're not algae, although you'll still see them called that sometimes, particularly if you go to a health food store. I guess people would rather eat blue-green algae than bacteria. But anyway, these guys are oxygenic. So what does oxygenic mean? They produce oxygen. So these are bacteria that produce oxygen. They do it the same way plants do. So oxygenic means oxygen generating or oxygen producing. It's not surprising that they produce oxygen the same way plants do, because the process of producing oxygen in plants is associated with photophosphorylation, and that involves organelles called chloroplasts, and chloroplasts were originally cyanobacteria. Remember that endosymbiotic theory. Well, anyway, so these things produce molecular oxygen. They do it in association with their photosynthetic activity. They do it by splitting water molecules. These things sometimes live symbiotically with lichens, so they can pair up with fungi and live as lichens, but a lot of them are free living. A lot of them live in fresh water. Some produce akinete, some produce heterocysts. They also sometimes produce sheaths, so they might be inside of a sheath, multiple cells inside like a cellophane wrapping. They tend to form these chains. These chains of cells are called trichomes. We're going to be looking at trichomes for a number of different cyanobacteria types in the laboratory. Notice that these appear to be green. Sometimes cyanobacteria are green, but they produce pigments called bacteria chlorophylls that are not necessarily green. So their photosynthetic pigments can be green or bluish or orange or brown. They come in a variety of different colors. There are some cyanobacteria that can cause intoxication. Now, typically when you see that term, people think alcoholic intoxication, because if you drink too much alcohol, you can experience intoxication. But intoxication just means you have consumed substances that are toxic. And cyanobacteria produce some substances called microcystins. These are peptipeptides, so they're little short peptide chains. Apparently the amino acids are put together not in association with ribosomes. So they make these microcystins, and they can cause severe liver damage, which I put that little bottle of blue-green algae on there just because I'm not sure which ones produce the microcystins that are hepatotoxins and can cause considerable damage. Are they the same ones that get packaged up and sold in the health food stores? I hope not, but I don't know. There are situations where cyanobacteria can be abundant in fresh water, and if cattle drink the water, it makes them very, very sick. Or if humans drink the water, it makes them very, very sick because of these hepatotoxins. Now, there are also phototrophic bacteria that are anoxygenic. If oxygenic means they produce oxygen or generate oxygen, anoxygenic means what? A or N in front of a word means without. So these are without oxygen production. Notice this group, I don't have a single phylum represented here. I just wanted to mention the anoxygenic phototrophic bacteria. In the box at the back of the laboratory, there's a plexiglass box back there, and if you look on the surface of it, you'll see patches of pink, patches of dark green, patches of black. Those organisms that are living in there are primarily anoxygenic phototrophic bacteria. So they don't produce oxygen, but they do use light as an energy source. The reason they are not capable of producing oxygen is because they can't split water molecules. These guys produce pigments called bacteria chlorophylls. And they can, these apparently can capture light, wavelengths of light that other organisms can't, that this light penetrates down into the mud. Although the ones in the box, they're getting the light through the plexiglass. There's a lot more growth on the side facing the room light than on the other side that's facing the wall. Now we're moving to the next phylum, the Proteobacteria. This phylum is named for the Greek god Proteus. Anybody familiar with Greek mythology? Maybe a little bit. Well, so Proteus was a god that could take on different forms. So he could often come down and be walking around in the town, and people would recognize him because he looked like somebody completely different. So in the phylum Proteobacteria, we have quite a wide range of shapes and sizes of the bacteria. This is a very large group, the Proteobacteria. They would be predominantly the ordinary gram-negatives, although there are quite a number of organisms that would also be bacteria with unusual properties that fit into this group. So we'll look at some specific examples. A couple of things to think about. As I'm going through these groups of bacteria, on next week's quiz, expect to see a matching situation where you're matching genus names or possibly phylum names, the phylum Dianthocus thermus, the phylum Cyanobacteria, where you're matching the name with important characteristics for the group. The terms that you might find on the define or fill in the blank would be terms like methanogenic and oxygenic and bacteriorhodopsin. Okay, so we're going through a lot of groups. Try not to panic about this. At least not yet. Okay, so our first group, the Rickettsia and Orientia. These are two genera. They are hypertrophs. So part of what we're doing today is going through some of the terms that were introduced in the taxonomy section. What does hypertroph mean? It means obligate intracellular parasites. So these are organisms that can only grow and reproduce when they're inside a cell. These are pathogens, at least sometimes. They get into mammalian hosts. So they grow in specific types of cells, the reticuloendothelial cells, which are in lymphatic tissue. Also the vascular endothelial, which are the cells that line the blood vessels, and in erythrocytes. They're transmitted typically by biting arthropods, not necessarily insects, because usually lice or ticks. And apparently what they do is cause the human cells to take them in through endocytosis. It would be phagocytosis because they're cellular in size. So here we see a cell that has ingested a whole bunch of these things. The bacteria are stained red, so you can see them inside there. And of course, if they're taken into a host cell, they're likely to cause disease symptoms, which they do. So Rickettsia rickettsii, these are the causative agents of Rocky Mountain spotted fever. Rickettsia prowazekii are the causative agents of epidemic typhus. And the third one, which is now called Orientia tsutsugamushi, these used to be Rickettsia tsutsugamushi, they cause a disease called scrub typhus. Apparently, Rickettsia prowazekii killed more people during World War I than did gunshot wounds, and it had to do with the conditions that on the battlefield where people dug these trenches, and they had all these soldiers in these trenches, and they were sitting there for months at a time. Terrible sanitation. They were all infested with lice, and then the Rickettsia got on board and made a whole bunch of people sick with epidemic typhus. The next genus of interest, again, we're still in the phylum Proteobacteria, is the genus Wolbachia. These are very interesting bacteria because they are residents within so many other organism types. It's thought now that Wolbachia might be the most abundant type of bacteria on the surface of the planet. The reason for this is because they're parasites of insects. Think about how many different types of insects there are. They're also parasites of protozoa, single-celled animal-like organisms. And they're parasites of roundworms, and undoubtedly other types of animals as well. What's interesting about Wolbachia, when they get into a host, they tend to become associated with reproductive tissue, and they have some really interesting effects on their host organism. This is an image of, I believe, a paramecium. So this is a single-celled animal-like organism, and those green spots are Wolbachia. So they're throughout that protozoan cell. So they tend to infect the ovaries and testes if they're getting into a multicellular organism. If they're inside a protozoan, there aren't ovaries and testes, they're just in there. But in multicellular organisms, they do interesting things relative to reproduction. So they can kill off all the males of a species, or they can feminize all the organisms in the species, basically cause males to become females, or cause males to become sterile so they can't mate. They can cause females to engage in parthenogenesis. Parthenogenesis is a process where female organisms give rise to other female organisms without mating. So pretty soon the entire population is female, and they can just continue to populate with female organisms. Or they can cause cytoplasmic incompatibility. So if you have one member of a species that is infected with one strain of Wolbachia, they can't mate with a different organism that's in the same species but has a different strain of Wolbachia. So it kind of separates populations and can lead to new species being developed. And this is talking about several arthropod species are so influenced by Wolbachia, they can't reproduce unless they have Wolbachia. So it was a parasite, but then, as I said, when you have symbiotic relationships, the most common type of symbiotic relationship is mutualism. The Wolbachia are benefiting, and apparently they've changed the population of the arthropods to the extent that they have to have the Wolbachia or they can't reproduce. These things infect various types of nematodes that are called filarial worms, and several of these are parasites of humans. So one, Dirofilaria immitis, actually this is the canine heartworm, so it's more likely to be a parasite of dogs, sometimes cats, but it can parasitize humans. Another one called Onchocerca volvulus, this is a parasite of humans that causes river blindness. And then another one, Wucheruria bancrofti, causes a disease called elephantiasis. So here we see some images. On the left-hand side, a heart with heartworms, an eye that's damaged by river blindness, and then elephantiasis. These nematodes get into the lymphatic system and cause the fluid to build up in the extremities, and you get these great big swollen limbs, swollen legs, and so forth. Well, it turns out that the symptoms of disease associated with infections with Dirofilaria or Onchocerca or Wucheruria have to do with Wolbachia. And so people are finding that if they can treat the people that are infected with these roundworm parasites, if they can treat them with antibiotics and kill the Wolbachia, then oftentimes that gets rid of the symptoms. And also the symptoms, the inflammatory response is more a response to Wolbachia than it is to the parasites. So this is fairly important findings, finding out what is Wolbachia doing and what might people be able to do to control it. The other thing they're looking at is because Wolbachia is associated with insects, a lot of insects are associated with diseases caused by protozoa, diseases caused by viruses. And so they're looking at how can they manipulate Wolbachia to change the insect life cycle so that the insects can't transmit the disease-causing agent. They've tried to modify the mosquitoes. I think this goes on to talk about it. We're very concerned about climate change because as the climate warms up, a lot of the parasites that I've just mentioned, the river blindness, the Wucheruria, not so much Dirofilaria because we have Dirofilaria here, but the other two are restricted to the tropics. As climate change warms the planet, the tropics are going to expand. And so this is something that people are very concerned about. A lot of diseases that are involving mosquito transmission, Zika virus, malaria, yellow fever, dengue fever. They're looking at what can they do to manipulate the insects, the mosquitoes, using Wolbachia. And so there's quite a bit of work going on here, specifically with Aedes aegypti, which is the type of mosquito that is the vector for dengue fever, chikungunya, and Zika. They can modify the Wolbachia that's in those mosquitoes, or maybe introduce a new strain of Wolbachia. They can shorten the insect's life cycle. They don't have time to grow the viruses. Then they can't transmit.
So I'm trying to do the best I can to follow you. Is Wolbachia a type of class in Protobacteria?
It's a genus.
Oh, it's a genus.
Most of these names that I'm putting up here now are genus names. If they're in italics, they're genus names. So Wolbachia is a genus. There are multiple different species. And as I said, if you can get antibiotics that would target Wolbachia, you might be able to eliminate the symptoms associated with some of the filarial infections, because it's really the Wolbachia that's causing the damage. The next genus, it's hard to tell those are italicized, but those aren't. So this is a genus, Rhizobium. Rhizobium is kind of overlooked, and very often plants get credit for fixing nitrogen. You hear about farmers planting alfalfa or clover or beans, some kind of cover crop. What they're doing is trying to improve the nitrogen content of their soil. It turns out the plants have nothing to do with nitrogen content of the soil. Well, as soon as I say that, I think, well, yeah, they do. The plants actually make conditions better for Rhizobium. So these are nitrogen-fixing bacteria. They can take molecular nitrogen, N2 from the air. They can convert it into nitrate, nitrite, or ammonia, and those forms of nitrogen the plants can use. These things are typically associated with plants that used to be called legumes. The family was Leguminaceae, and for some reason, recently, that family name has been changed. And it's kind of too bad, because legume, which was the common name for beans, peas, alfalfa, vetch, legume is part of a term relative to what the plants produce. So here we see root nodules. These are little structures on the surface of roots. These little root nodules. The plants form those, and Rhizobium lives in them. So Rhizobium is a symbiont, lives with legumes. The plants produce a substance called leg hemoglobin. Leg from legume. But what's hemoglobin? I know you guys know this. We have hemoglobin, right?
Yeah.
It's in our red blood cells, yes? It is a transporter for blood gases. It can pick up oxygen, it can also pick up CO2. If it's carrying oxygen, it's oxyhemoglobin. If it's carrying carbon dioxide, it's carboxyhemoglobin. But hemoglobin is this red-colored pigment. It's a protein that can transport gases. It turns out leg hemoglobin, the leg, again, it's produced by legumes, has a very similar function. It can pick up oxygen. It is believed to create an atmosphere inside the root nodule that improves the efficiency of Rhizobium. But it's possible, because Rhizobium is a respiratory type bacteria, the leg hemoglobin might be transferring oxygen to the Rhizobium. At any rate, they live better inside those nodules, and if you cut those things open, so here you can see root nodules, kind of a close-up view. Notice how they're pink? If you cut these open, they are red inside. Red, like red blood cells, like hemoglobin, but in this case it's leg hemoglobin, and it's made by these leguminous plants. So again, nitrogen fixers are bacteria, not plants. Clover, alfalfa, beans, they can't fix nitrogen, but bacteria can. And Rhizobium play a critical role in the nitrogen cycle. They're taking N2 molecular nitrogen, putting it into the soil in a form that plants can use. So this improves the soil. This is a big deal, because farmers that are trying to grow plants on soil that's poor spend a lot of money on fertilizer. They're having to put nitrogen-containing fertilizer out. If they could get the plants to fix their own nitrogen, the plants can't. But if they could get the plants to live symbiotically with Rhizobium or some other type of bacteria that could fix nitrogen, then they wouldn't have to use fertilizer. Another type of bacteria that fixes nitrogen is called Agrobacterium. Agrobacterium tumefaciens is of interest because they are nitrogen fixers and they are plant parasites. And they apparently produce these, well, they cause a disease on plants that's called crown gall disease. If you look at the back of the room on top of the cabinet, you see those roots with the balls on them? I don't want to walk back there. But anyway, those are galls made by Agrobacterium tumefaciens. So it's kind of like the root nodules that are formed by the plant to grow rhizobium, but these are a lot bigger, and they're really hard, woody structures. The Agrobacterium apparently causes the formation of those things, and they do it by transferring plasmids into the host plant. Well, so investigators recognized this, that Agrobacterium has these plasmids that can be transferred into plants, and then investigators can use these plasmids to transfer genes into plants. And that leads us then to biotechnology and the production of genetically modified organisms. What they've done is take out the genes that would cause damage to the plant, but they can use the plasmids to put genes into the plant depending on what they want the plant to do. So again, if you could get the plant to maybe accept something like rhizobium, or accept some other nitrogen fixer, then the plant might be able to grow without fertilizer. But more commonly, what's happening is food plants are being genetically modified in ways that maybe as consumers, we're not going to be real happy about. So for example, there's a substance, well, how many of you heard of GMOs? Surely, because they've been in the news. So GMO is just genetically modified organisms. Humans have been genetically modifying food crops for centuries because we've been doing genetic crosses and trying to get the best food crops. But now what's going on is genes are actually being put in that allow plants to be resistant to chemicals. Specifically, there's a chemical glyphosate, which is in Roundup. Roundup is an herbicide that's used to kill weeds where crop plants are planted. The plants have been genetically modified, so they're not damaged by the Roundup, so farmers can spray that stuff over and over and over again. And now what we're finding out is glyphosate is a carcinogen. And so how much of our food crop is contaminated with a carcinogen because the plants are genetically modified? Not a good thing. There are some plants that are genetically modified to produce insecticides that kill insects. This was good in terms of controlling pest insects, but it's not selective. And so we're seeing a loss of a lot of butterfly species because these genetically modified plants are killing them. So there's a downside to genetically modified organisms. Another organism type that is a nitrogen fixer is Azotobacter. This is one of the ones that we were doing our enrichment for. So they can fix nitrogen. Again, they take molecular nitrogen from the air and they can convert it to nitrate, nitrite, or ammonia, which then the plants can use to build organic compounds. Of course, the bacteria can too. You're going to get to see Azotobacter in the lab tonight. So the next genus, again, we're still in the Proteobacteria group. The next genus is Spherotilus. The Spherotilus organisms are not significant as pathogens. They don't live symbiotically with other organisms, but what they do is they build these tube-like sheaths, or straw-like. They live inside these sheaths in water, and the sheaths almost look like underwater spider webs. You can see some images of the sheaths with the bacteria in them. You really can't see the sheaths very well if the cells aren't there. So here you see a lot of cells inside the sheath, and the sheath is nearly transparent. In the water, they make these mats of fibrous material, and they can block water flow quite successfully. So these become problematic when they get into slow-moving water, slow-moving streams, or fish tank filters. They'll block the water flow, or well pumps. Apparently this is one of the places where they really cause problems because they get into well pumps and gum them up so that the pump doesn't work anymore. Can't move the water against all that sheath material. So that's Spherotilus. Interesting little bacteria that live inside of sheaths. The next genus is Thiobacillus. These are chemoautotrophs, so these are organisms that are using inorganic compounds for both carbon and energy. Sometimes they're called lithotrophs. Litho refers to the earth, so they're basically using rocks, minerals, soil, that kind of thing, as their nutrient source. And they're involved in what are called biogeochemical cycles, iron cycles, sulfur cycles, etc. They play a very important role in the environment by cycling minerals. A specific species, Thiobacillus ferrooxidans, can use hydrogen sulfide as an energy source. So they're able to pull electrons and hydrogen protons off of hydrogen sulfide. As a result, they produce sulfuric acid as a byproduct. Well, sulfuric acid can cause minerals to be leached out of poor grade ore, so they use these guys for mining operations and get Thiobacillus ferrooxidans established. As they're living in the low grade ore and they're producing sulfuric acid, it will leach the minerals out, and then the minerals can be captured. So they're using them for mining. They're also supposed to be used to remove sulfur from coal, supposedly to produce coal that will burn clean. No coal burns clean. The next genus is Neisseria. Most important aspect of Neisseria, there are some important human pathogens in this group. So Neisseria gonorrhoeae causes gonorrhea. Neisseria meningitidis causes meningitis. But we often have Neisseria species living in our throats. These are typically gram-negative cocci in pairs. One of the things you'll notice about these, when you have cocci in pairs, a lot of times where the cells join, they have a flat side. So they're cocci, but cocci that are kind of mashed into one another. They are often part of our normal microbiota in our pharynx, a lot of different species. The ones that are part of your normal microbiota would not be pathogens, but they probably would protect you from something like Neisseria meningitidis. The next genus is Pseudomonas. This is another one that we were doing an enrichment for. So Pseudomonas. There are some pathogens in this group. Some of them are infective agents of plants. Some are infective agents of animals. For us, Pseudomonas aeruginosa is the most significant because it is an important human pathogen. More importantly, what we were taking advantage of when we did our enrichment, we were looking at the fact that these bacteria can use unusual organic compounds as their source of carbon and energy. Some of them can use a very wide range, like 200 or more different types of chemicals. Because they can break down weird organic compounds, they're used in bioremediation. How many of you know what bioremediation is? In the simplest terms, it's environmental cleanup using biological agents. So bioremediation, basically what it means is if you have some kind of toxic substance you want to get rid of, you feed it to bacteria. There are some fungi that are used this way also, but Pseudomonas is used quite extensively for bioremediation. And in the state of California, this is very significant because if you're a company that is producing toxic waste, that toxic waste is yours. You can't just dump it in the environment. It's illegal. You have to contain it or get rid of it, as in break it down. These guys will break it down. And so there are regions that have been set aside where the toxic substance can be put out with some nutrient material, put the Pseudomonas on it, and let them eat it, let them break it down. And they do a pretty good job. Unfortunately, sometimes they break down things that we'd rather they didn't, so they can break down petroleum. And there was a story about Pseudomonas getting released onto a parking lot during a rainstorm, and when the rain stopped, all the asphalt on the parking lot was gone because they ate it, and there was nothing left of that. So they were very efficient. In that situation, it's not so good. One of the organisms that's used in bioremediation is called Pseudomonas fluorescens. They're called this because they produce a water-soluble fluorescent pigment. We have Pseudomonas fluorescens as one of our morphological unknowns. If it turns out you have that organism, see me about getting the ultraviolet light out, because you won't see the fluorescence without putting a UV on it. But if you shine it on there, it produces this kind of greenish or bluish fluorescence. These can use sodium benzoate. So in our Pseudomonas enrichment, sodium benzoate was the only organic compound. You give them that, they can eat it, even though it's toxic to most bacteria. Another type of Pseudomonas is Pseudomonas syringae. Now these are recognized as being plant pathogens, but for some reason they produce a protein that is used as a nucleating protein for snowmaking operations. So where snow is being produced, you have to have something to get the ice crystals to form around. So they put the protein from Pseudomonas syringae into these snowmaking machines and produce quantities of snow. Pseudomonas aeruginosa is the pathogen. These guys produce a pigment called piocyanin, which is this green color. It causes the medium the organisms are growing on to turn green. And the stuff is toxic to host cells. So here we see Pseudomonas aeruginosa in a wound, this green color. Here it is on a plate, you see the green color. Their ability to produce the piocyanin, to produce virulence factors, is dependent on quorum sensitive. So if you have just a few Pseudomonas, they're not going to do anything. When you have a lot of Pseudomonas, they're going to start producing virulence factors and cause a lot of damage. They actually inhibit protein synthesis, so the wound can't heal. Next important genus is Photobacterium. Photobacterium, well, what does photo refer to if you're not talking photography? Anybody know? Light. So Photobacterium, these are organisms that have something to do with light. Actually, they are bioluminescent. They often live symbiotically with deep sea organisms, fish, or squids. A lot of these organisms have what are called light organs, and they appear to produce light. Again, it's not the fish or the squid that's producing the light, it's Photobacterium. There are also some vibrios that can do this. Technically, this light production is called bioluminescence. So Photobacterium are bioluminescent. They use some substances called luciferins, and then they act upon the luciferins with enzymes called luciferase enzymes. The root word there, lucifer, means light bringer. So luciferins and luciferase enzymes are used to produce light by Photobacterium. And this is the kind of light that you can see. In order to see this, you have to be in a dark room. Later on this semester, you'll be able to see light production by Photobacterium. We have Photobacterium phosphoreum that was collected from a red snapper. Now these guys, as I said, they're living symbiotically with animals. The animals benefit because they can use the light. You've seen the movie Finding Nemo. Some of them have light on the end of a little stalk and they can attract prey. But the bacteria also benefit because they have a place to live. A lot of times these light organs, they're just a surface patch of skin. Maybe there's a patch of skin that can cover it up so the lights can be turned on and off. But they have nutrient available to them there. So and sometimes they live inside the fish as well. They can live in the GI tract. So these light organs appear to turn on and off. They can have different distributions on the surface of the fish. And also the light can be different colors. They're not all exactly the same. The one we have is that kind of light blue or greenish blue. There are genes that encode the proteins associated with light production, the luciferins and the luciferase enzymes, and the genes are called lux genes. Lux also means light. These genes are regulated through quorum sensing, and there was a period of time where researchers wanted to use lux genes. They could genetically modify some organism, and they wanted to be able to trace disease processes through a host organism by watching the light. Supposedly, they infected tobacco plants and developed tobacco that would light by itself. Well, anyway. The other thing about Photobacterium, apparently they're very sensitive to pollution, so they can use them to detect pollution in water. If you have them lighting up, they're producing their light, and then the water becomes polluted, the lights go out. So they can be used to test water for pollution levels. The next major genus is Escherichia. So probably you more commonly hear of these as E. coli. The E stands for Escherichia. So E. coli and friends. This is a group of organisms that we use in our physiological unknown 1. There's a group, they all belong to the order Enterobacteriales. They're relatively closely related. And they're part of our normal microbiota. So a lot of times they live in the gut, although some of them they live in the guts of other organisms, or sometimes they live in water or soil. At any rate, you're going to see these again when we do physiological unknown 1. So Salmonella, Shigella, Enterobacter, Serratia, Proteus, Klebsiella, Citrobacter, Yersinia. Actually, you're not going to see Yersinia or Shigella because those are pretty nasty pathogens. Yersinia causes bubonic plague, and Shigella causes dysentery. We try not to give students those. But you will work with Escherichia coli. So Escherichia coli was named for Theodore Escherich. He discovered these bacteria back in 1885. Since then, anytime somebody wanted to work with bacteria, they knew they had these bacteria because they live in the gut. So they could get their own Escherichia out and play with them. Consequently, they're some of the best studied organisms on the planet. There are some that are important human pathogens. The strain O157H7 was responsible for labels on ground beef telling you don't eat it raw. O157H7 can cause hemolytic uremic syndrome, which kills people. You shouldn't eat raw meat anyway. There's other things in there. But Escherichia coli has multiple uses. So it is used as an organism for water testing. Later in the semester, we're going to do the bacteriological examination of water. Turns out E. coli is what we're testing for. So if you want to know, is your water contaminated? You're testing for E. coli. Why? Because they live in the gut. They don't normally live out in vegetation. If they're in water, it means there's gut contamination to that water. They're also used in research situations, predominantly genetic research, again because the genetics of Escherichia have been more thoroughly studied over time, and also because they carry some important viruses. We've learned a lot about genetics by studying viruses. More recently, Escherichia strains have been genetically modified themselves to produce what are called recombinant proteins. This means you can take genes from other organisms and put them into E. coli, and E. coli will produce proteins. There are some important human proteins, for example, human growth hormone, factor VIII, which is a clotting factor, human insulin. These are now produced by Escherichia coli. So the bacteria producing them is way cheaper. You can't take these chemicals out of humans. It's like, no, you can't sacrifice a few humans and take their pancreas to get human insulin. And we used to use pig insulin, but it didn't work as well. So now you can use human insulin that's produced by E. coli. It's pretty effective. The next genus is Delovibrio. The B on this word is silent. The name for this organism means comma-shaped leech. So Delo, or Della, means leech, and Vibrio means a comma-shaped organism. They're like a short rod, but they're bent. What's interesting about these organisms is that they infect, they kill other bacteria. We usually think of bacteria infecting humans or infected plants. These guys attack other bacteria. They were initially thought to be hypertrophs. It was thought they went into the host cell, and then additional research said, no, they don't. They just go into the periplasmic space. So here you see a host cell, which is Pseudomonas. This is the Pseudomonas cell. Here's Delovibrio, this little comma shape. They are flagellated, and it's just touching the end of that cell. It's going to go through the outer membrane and into the periplasmic space. And it's going to reside there and produce enzymes that basically kill the host cell, and then it's going to feed on it. So you see the life cycle. Delovibrio approaching the host, touching the host, getting inside the host. The host cell you can't tell what's going on there for a while, but ultimately the host cell is going to start releasing more Delovibrio, and a whole bunch of them come out. So one goes in, a multitude come out. That's where they reproduce, taking the nutrient of their host. The next phylum is the phylum Firmicutes. Again, never mind the number. These are ordinary gram-positive bacteria. So the proteobacteria were ordinary gram-negative, and some unusual gram-negative. These are ordinary gram-positives. The first important genus is Clostridium. Now, Clostridium is somewhat unique because they're anaerobic. What does anaerobic mean?
Doesn't require oxygen.
Not only does it require oxygen, but oftentimes can't tolerate oxygen. If an organism is an obligate anaerobe, they can only grow and reproduce where oxygen isn't present, because oxygen will tend to be toxic to them. These are endospore forming. We don't grow them in this laboratory because we're growing plates in either the incubator or in your lab drawer. They're always exposed to the air. And Clostridium is anaerobic, so you're not going to see them growing. Oh, there they are growing. There are several species that are significant pathogens. So Clostridium tetani is the causative agent of tetanus or lockjaw. This organism is not particularly invasive, but it gets in with a puncture wound and then it produces toxins that cause tetanus or sustained muscle contraction. Clostridium botulinum, closely related, produces the botulism toxin. Botulism toxin is ingested, and then that toxin can cause flaccid paralysis. And Clostridium perfringens causes gas gangrene. This is more problematic where people are exposed to shrapnel and get a lot of dirt in a wound. Their tissue's all torn up. Clostridium perfringens can get established and cause considerable damage. And Clostridium difficile, more commonly referred to as C. diff. This is a gastrointestinal Clostridium. Kind of an interesting sidelight about C. diff. Clostridium difficile infection has become increasingly common, probably because many people are on long-term antibiotics. If you're taking antibiotics for a long period of time, you're messing up your normal microbiota. Apparently Clostridium difficile lives in the gut, it's in the human gut. If you take away their competition, they can begin to grow out of control and they can cause severe damage to the gut wall. They can kill people. That's not a nice infection. The problem is you get Clostridium difficile infection, C. diff infection, from taking antibiotics. So it's tough to control with antibiotics. So what do you do about it? There's a new procedure called repopulation. Repopulation means basically giving somebody poop from an individual that's healthy and reestablishing normal microbiota within their gut. Exactly how you get that in there is variable.
Through an NG tube.
Yeah.
It's a fecal transplant.
Fecal transplant. Yeah, more commonly called a fecal transplant than repopulation. I think that was some microbiologist. And there are actually people that have been kind of cataloged. This person has good, healthy gut microbiota. They can donate poop. They won't take it from just anybody. Okay, next genus, Mycoplasma. Again, we're in the phylum Firmicutes. These are gram-positive. Mycoplasma are tiny little bacteria. So look at the size on that, 0.1 to 0.25 microns in diameter. You couldn't see those things with a light microscope. They're just too little. They were initially thought to be gram-negative because apparently they don't have peptidoglycan in their walls, but genetically they're gram-positive. So they're in the phylum Firmicutes. They little bitty bacteria, extremely sensitive to osmotic pressure because they don't have the peptidoglycan walls to protect them. And some of these are associated with human disease. The term pleomorphic applies to bacteria changing shape. If bacteria are pleomorphic, some of them are cocci, some of them are rods. These guys are going to be quite flexible without the peptidoglycan. But again, sensitive to osmotic pressure. And they're responsible for causing what was sometimes called non-bacterial pneumonia or walking pneumonias. People didn't know bacteria were causing it because they couldn't find the mycoplasma. They were too little. Mycoplasma pneumoniae causes walking pneumonia. The next genus is Bacillus. Of course, we know that's a shape too. But the Bacillus organisms, Bacillus is an important genus. These are either aerobic or facultatively anaerobic. If they're aerobic, they require oxygen. If they're facultative, what does that mean?
Either way.
Either way, it means they can live with or without oxygen in their environment. Lots of bacteria are facultative, and bacillus are extremely common. This genus, as new strains of bacteria were identified over time, it became unwieldy. It was just too big, and so it's been split. Now there are bacillus, and then there's Paenibacillus, Lysinobacillus, Brevibacillus. They go on and on and on. Related organisms. So some of the information I have about bacillus being of particular interest, sometimes it's bacillus and sometimes it's one of those related types. So Bacillus thuringiensis, this is an important organism type, that particular species. BT stands for Bacillus thuringiensis. And you can buy a product, if you go to a nursery, you can buy BT, another trade name is Dipel. This is Bacillus thuringiensis, and what it's used for is to kill insects, pests on plants. It can be used to kill mosquitoes in water as well. You buy Dipel or BT. If you put it on plants, what it's going to kill are the insects that produce butterflies and moths. So the little cabbage butterflies that eat up your plants, you put BT on there and it kills them. But if you put it in water, it'll kill mosquitoes. Then one that used to be Bacillus sphaericus is now Lycaena Bacillus sphaericus. These are also used to kill mosquitoes in water habitats. And another one, Paenibacillus larvae. These kill bees, so they cause what's called American foul brood in bees. This is a very destructive disease for beekeepers. It's quite problematic. And then there's Bacillus anthracis. We learned about those guys. They're the causative agents of anthrax. They were the ones Koch was working with when he developed Koch's postulates. But here's an interesting sideline. Bacillus anthracis, Bacillus thuringiensis, and Bacillus cereus. And now there's some others in this same category. They're called the Bacillus cereus group. Turns out these bacteria are all the same organisms, except different ones have plasmids. So if they're Bacillus anthracis, they have plasmids that carry virulence factors. If they're Bacillus thuringiensis, they have plasmids that allow them to produce these chemicals that kill insects. Bacillus cereus doesn't do either of those things. So they're all very closely related. Here we see an anthrax infection. Bacillus subtilis. This is the bacillus that's responsible for producing bacitracin. This is an antibiotic, oftentimes used as a topical antibiotic in ointment, bacitracin. And then Paenibacillus polymixa, used to be Bacillus polymixa, is the organisms that produce polymyxin. Polymyxin is another antibiotic. We're not going to get through the prokaryotic cells, so we're going to stop. But that's okay. So next time we'll finish up prokaryotic cells, starting with Staphylococcus. So go ahead and take a break. We'll come back and have lecture quiz 3.